All right, good afternoon, everybody. First, I would like to thank the ISMB Selection Committee for giving me the opportunity to speak here today. And I'm a PhD candidate at the University of North Carolina at Charlotte, and my PI is Dr. Junda Go. And today I'll be talking about structure-based predictions of transcription factor binding specificity using an integrative energy function. So all day we've been talking about transcription factors, and we know that transcription factors bind to some transcription factor binding site to regulate genes, whether it's in the promoter region or the enhancer region. But why do we study transcription factor binding sites? Well, our group does it because we want to understand how a transcription factor scrolls along the DNA, finds its binding site long enough so that transcription can begin and then continues going. When we understand this, then we have more information so we can expand the annotation of our genomes. And once you have a better gene network, then we can understand how these aberrations to the gene networks may lead to genetic disorders, such as cancers. Also, when we understand the mechanism of how transcription factors bind to DNA, we can start looking into things like re-engineering transcription factor specificity, which is actually being done in zinc fingers right now. And once we understand this, then we have a little opportunity to expand or to build more rational drug design. So the traditional methods of deciphering transcription factor binding sites are, we have our classical methods, which are our gel-based methods, such as DNase 1 splitting and gel mobility shift assay. But these methods are pretty much outdated now, and they're very time-consuming and not suitable for large-scale experiments. So in the last decade or so, there's been a boost in high-throughput experimental methods, such as CLIX, your chip-based methods, and your protein-binding microarrays. And while these are great for doing thousands of experiments at the same time, they still don't show us how these transcription factors actually bind to the DNA. So a lot of computational methods have been derived or developed in the last few years. We have our sequence-based methods, which are computationally efficient, pretty quick. They're accurate for a specific family as they are trained for a specific family. However, sometimes they are limited to that particular family, and many times you have a high false positive rate. The other methods are our structure-based methods, which we believe that there are more information in structures, which actually gives us the opportunity to possibly look into inference into the mechanism. We can actually now describe how it actually works. And these methods are possibly generalizable. However, because of the complexity of some of these methods, they can be computationally expensive, and the most difficult problem is they require higher resolution structures, which are hard to come by. But these methods can actually explain to us how and why transcription factors bind there, whereas the other methods only explain when or where. So how structure-based methods of transcription factor binding site prediction done? Well, typically, we'll get some high-resolution structure and permutate your DNA sequence to all the possible permutations of that sequence of the 4 bases. You'll apply some energy function to it, and you'll get energy scores for all your sequences, have some distribution, and have some criteria to select your highest scoring sequences to come up with your binding site. And typically, that's displayed as a binding motif or binding logo. So a few years ago in our lab, we showed that some may argue that depending on your sequence of your DNA, you may change the shape. So we looked at the different, the RMSD and of the DNAs bound to the recognition helix of some domains. And we found that your transcription factors typically will bend or manipulate the shape so that your DNA bound to your recognition helix typically have the same shape for that structure, for that protein. So we use that assumption into our algorithm that even though we change the sequence of the DNA, it wouldn't change the shape severely enough to affect the binding calculations, the binding energy calculations. After changing the DNA, now we apply our energy functions. And typically, we have 2 major types of energy functions. We have our physics-based energy functions, which comprises of our bonded terms, your hydrogen bond, your bond's length, your bond angles, your torsion angles, and your non-covalent terms, which comprises of your electrostatic energy and your van der Waals terms. Or you have your knowledge-based energy functions, which are basically a distance-based function, whether it's a residue-based, knowledge-based function, or a all-atom. It basically takes a distance between a particular amino acid or base, and depending on that probability of having that amino acid within that distance of that base, you can convert that to energy using the Boltzmann distribution. But again, both of these methods have their pros and cons. For example, your physics-based, you may be able to explain your mechanism. However, force fields are not well understood. So while conceptually you should be able to, it should be the best energy function, since we don't understand some aspects of the force field. For example, our hydrogen bonds, they require, while they're initially electrostatic, they have some dependency on angle and distance, which isn't fully understood, but we have multiple models for it. And also, we don't fully understand the whole hydrophobic effect. So these little aberrations, or these falling short in our knowledge of the physics-based potentials, makes things a little difficult, and as a result, we have inaccurate predictions. Plus, doing these complex calculations on thousands of structures is computationally expensive. While our knowledge-based potentials, they're much less computationally expensive and they perform relatively well, they still need some improvement because some forces the mean force may actually misrepresent some interactions. And what do I mean by mean force? Well, for example, we have lysine and arginine, and they tend to form specific hydrogen bonds inside in the major groove of your transcription factor or your binding site or the DNA major groove. So what occurs a lot is that we have non-specific interactions with the minor groove, and since these amino acids are basic, so they're positively charged, they tend to also to interact with the phosphate backbone, which is negatively charged. As a result, the specific hydrogen bonds are drowned out by the non-specific interactions, and we can't get enough signal in some of the knowledge-based potentials, to this. So a former student in the lab who actually just graduated this past May, she looked at the propensity of some amino acids in between different classes of transcription factors and DNA bases. And what she found was that, as we expect, the typical polar amino acids, which form hydrogen bonds, all have high propensities for your highly specific DNA binding proteins, such as your structural enzymes, and your multi-specific binding proteins, which are like your transcription factors. And as I just mentioned, your arginines and your lysines, they also have their high propensities for your non-specific interactions or your non-specific DNA binding proteins, like your polymerases. But something that stood out to us was that tyrosine, which is pretty much, well, it's amphipathic, but it's also, it has this huge aromatic ring, and throughout the literature, there's studies that show that aromatic compounds are typically used for stability. They had a higher propensity before highly specific and multi-specific protein DNA binding, or DNA binding proteins as well. So since statistics basically drive hypotheses, we decided to look into this. And just an overview in case anybody doesn't understand or aren't very acquainted with how these aromatic systems work, is in pi interactions or in aromatic compounds, we have these pi orbitals. And when we have contiguous pi orbitals, the electrons can be delocalized and jump from orbital to orbital, forming some sort of an electron cloud. These electron clouds are now pulling the electrons from the hydrogens on the edges, and these edges are becoming more positively charged, and these electron clouds are in the center are more negatively charged. So now you can have pi-second interactions, which are predominantly electronic in nature. These interactions could be stacked, which is actually electronically unfavored because you have very negatively charges stacked together, or they could be T-shaped, where you have a positively charged edge interacting with a negatively charged electron cloud, or your parallel displaced, which is actually the most favorable one type of these interactions, where you have a positively charged edge interacting with each other's negatively charged cloud. So why do we care about this with protein DNA interactions? Well, the bases are all aromatic, and also because of the positions of the nitrogens and the oxygens and how they're distributed throughout the base, it actually changes the electronic density, hence changing the electronic landscape in the major groove. And our hypothesis was that this difference in electronic landscape in the major groove did affect the specificity, or actually contributed to specificity, because the electron clouds of the amino acids are all pretty negative. So in cytosine, for example, we have a predominantly positively charged, blue represents positive, predominantly positively charged area or landscape in the major groove, whereas guanine is predominantly negatively charged. Thymine, we have this little gray region, which is the methyl group, which is actually not a part of the pi system, so it's neutral in the major groove. And adenine, we have a distribution of positively and negatively charged. Now, these differences allow for specificity, which you can calculate using just an adaptation of Coulomb's law and summing the electrostatic energies. So what we've done was we know that these, or we assume, we hypothesize that these interactions, these pi interactions, can contribute to specificity, but due to a low count problem, since they don't exist as often as hydrogen bonds or other interactions in the limited protein DNA complexes that we do currently have in the PDB, these signals, or they are not adequately captured by the current knowledge base potentials. So what we did was we decided to combine or supplement our knowledge base potential, which is a residue level knowledge base potential between, so it measures the distance between the beta carbon of an amino acid and the centroid of a triplet of 3 nucleotides, and we call it our multibody potential. We supplemented this potential with a hydrogen bond energy term, where we calculate the explicit hydrogen bond energy, and a pi energy term, where we calculated the pi energy as just described. And the reason we add the hydrogen bond is so we can actually pull out the specific hydrogen bond information, as I described earlier, that they were submerged due to the mean force. So we combine these interactions and call this our integrative energy function. And as described before, we take our more significantly, or our higher, our better energies, sequences better energies, and we develop the binding load. Now we apply this to a data set of 29 non-redundant PDB structures, transcription factor binding structures that actually has an experimentally annotated binding site in JASPAR. And we show that for the majority in general, it either improves the prediction or it's pretty much the same, doesn't do a significant improvement. So in this graph, it's a bit small, but your blue represents your integrative energy, your red represents our multibody energy, and we also compared it with an all-atom energy function called DDNA3. Overall, there's an improvement, and we see that both visually and statistically. We use average Kullback-Leibler to do the average Kullback-Leibler divergence to do the statistical analysis, where it compares two PWMs, position weight matrices, and basically the lower the score, the better the match. So we looked at all 29 structures, and then we decided to look at some specific families. So first we looked at zinc fingers, and we saw that, at least visually and statistically, I didn't show the data here, it does do a little improvement, a bit of an improvement over the multibody function without the other atomic terms and the DDNA3 energy function. However, one problem with the analyses I've showed so far is that we do not, some of the binding sites actually have, or in JASPAR, represent binding sites for multiple domains on a bigger protein. So in aligning the predicted and the experimental binding sites and doing the comparison, we get a lot of noise from the flanking regions, which may actually be binding sites for other domains. So we zoomed in our study at Homodomains, which is the second largest transcription factor family in eukaryotes. And the reason we did this was because they've been well studied, and we know that the binding site is some variation of TAAT plus flanking regions. So knowing this, it's easier for us to identify exactly where the binding site is on the JASPAR logo. That being said, we found 11 non-redundant homodomains in the PDB, which had a corresponding binding site. And we did a similar analysis, and we found that The integrative energy function outperforms both methods quite well using, well, when we just looked at the core binding sites. We used information content weighted Pearson's correlation to look at the whole data set and look at the overall improvement. And we found that at least over 90% of the columns, which represents a particular predicted pace in our binding sites, has a Pearson's correlation of over 0.5. And about 80% or about 75%, around 80%, 75% has about a Pearson's correlation coefficient of 0.8, which is a little improvement over, the blue is integrative energy, so it has an improvement over the red, which is the multibody, and the green, which is the all-atom DDNA3. Then we looked at the individual proteins, and we found that about 80%, or a little bit less than 80%, of the proteins had 100% of the bases predicted correctly, and 80%, and 80% of the proteins had 100%, so it's kind of flipped. Whereas 65%, when we use the multibody potential, 65% of the proteins had 100% of the bases predicted correctly, and only 20%, you were using DDA3, had 100% of the bases predicted correctly. One thing I want to mention is that we did notice that DDNA3 actually did perform a little better on zinc fingers, so maybe an issue of it just doesn't perform extremely well on homodomains, but it does perform well on other transcription factor families. So we looked deeper to see what aspects of the energy function were contributing to the improve in performance. So we looked at 3 of the homodomains. These are 3 I'm just going to show examples of. And for example, in this one, 1B8IB, we see that when you add a hydrogen term, it improves the prediction over the multibody term. And that is because if you look at the structure, there's a arginine, again, interacting with this guanine, forming these hydrogen bonds, which are captured because of the explicit term, but it's lost when we just do the knowledge base term due to the mean force. Also, similarly with 1IC8, with the conserved cytosine, it's lost in the multibody and is not there when you have the bi-interaction term. But if you just add the hydrogen bond term, it's captured, and that's because your lysine is interacting with the complement guanine, the guanine on the complement strand, hence capturing this in our potential. And again, in 3RKQ, we find that this guanine is conserved because we capture this pi interaction, and the blue represents positively charged atoms, and the red represents your negatively charged atoms, and it captures this in our function. So our ongoing work will want to optimize this for large proteins. Our function didn't work very well for multi-domain proteins or proteins with excessive coils. So we want to optimize it for that, and we're actually expanding this to homology models. So we're developing homology models and applying our integrative energy function to it and testing that. And we actually are trying to make this algorithm a bit more efficient. So in summary, we have created an integrative energy function that combines a statistical potential, an explicit hydrogen bond term, and pi interaction term. Our results show that pi interactions actually help in transcription factor DNA specificity, which has not been seen before, or we have seen pi interactions there, but mainly for stability, but now we're showing that it's also important for specificity, and the integrative energy improves transcription factor binding site prediction, especially for smaller transcription factor domains. So I would like to thank, again, the ISMB selection committee for giving me the opportunity to share my research. My PI, Dr. Chintago, this guy, Dr. Takeda, who is the past postdoc in our lab, Rosario, who just graduated, and John, who was a summer student who helped us do some of the work. This work was funded by NIH, NSF, and GAN. So I have a poster if you want to talk about this some more, and also how I adapted this algorithm for homology models. I can describe that a little bit deeper. So thank you.